All right, today we're diving into something really on the cutting edge of cancer research. We're on the hunt for these hidden switches, the things that control some of the absolute deadliest diseases out there. It's a question that's haunted doctors and scientists for decades, right? Why are some cancers so incredibly hard to beat? I mean, you see a tumor shrink, you think you're winning, and then it just comes roaring back, sometimes even stronger. So what's going on? What gives cancer this terrifying resilience? Well, it turns out the answer might be hiding inside of a very special and very, very dangerous kind of cell. So let's meet the supervillains of this story. Because inside pretty much every tumor, there's this small elite group of cells. They're the real masterminds behind the whole operation. They're the true root of why cancer is so tough. And scientists have a name for them: cancer stem cells. Okay, so here's the best way to think about it. A tumor isn't just a big blob of identical bad cells. It's more like a weed in your garden. You can mow it down, cut the top off, but if you don't pull out the root, it's just going to grow right back. And that's exactly what cancer stem cells are. They are that root. They're the seeds that can regrow an entire tumor, even if only a few of them survive a treatment. And these things, they have actual superpowers. First, they can self-renew, which means they can make endless copies of themselves, an infinite supply of those seeds. Then they differentiate, meaning they spawn all the other cells that make up the bulk of the tumor. And this is the really scary part. They are resistant to therapy. Chemo and radiation, yeah, that might wipe out the regular worker cells, but these supervillain cells, the leaders, they often just shrug it off, completely untouched. And if that wasn't enough, they're also the ones that cause metastasis. They can break off, travel through your body, and start brand new tumors somewhere else. Okay, so if these cells are the supervillains, how in the world do we stop them? Well, to beat a mastermind, you need to get your hands on their playbook, right? Their instruction manual. And amazingly, researchers found it in a place they'd been overlooking for years, what they used to call the dark matter of our genome. So this really is the key question. What's actually controlling these things? What's giving them their marching orders? What's the hidden switch that turns on all those dangerous powers? And the answer, it turns out, wasn't in the genes we all know about, but in the vast, mysterious stretches of our DNA in between those genes. Get this, a whopping 80%, 80% of the stuff our genome makes, it doesn't actually build proteins. For the longest time, scientists just kind of dismissed this stuff. They called it junk DNA, just evolutionary leftovers. But wow, were they wrong. We now know it's anything but junk. Okay, so let me break this down with an analogy. Imagine your DNA is a massive library of blueprints. Now, what we call coding RNA is like taking one specific blueprint, say for a door handle, and giving it to a worker to build that one thing. Simple. But non-coding RNA, or ncRNA, that's the foreman on the job site. The foreman doesn't build anything himself, right? No. He's the one walking around telling the entire crew which blueprints to use, when to use them, and how many to make. He's the master regulator, the guy in charge of the whole operation. Okay, so let's put the pieces together. We've got our supervillain cells, the cancer stem cells, and we've got our foreman, the non-coding RNA. Now let's look at the playbook and see exactly how this foreman is controlling those supervillains. So what you're looking at here is basically the control panel. Right there in the middle, you've got the cancer stem cells, and all around them you see these different types of non-coding RNA. And you can think of each one like a little dimmer switch on a light. They're constantly fine-tuning all those superpowers we were just talking about, turning up the chemo resistance here, dialing down the self-renewal there. It's all being controlled. Let's take a closer look at just one of these switches. Let's talk about chemotherapy resistance. There's a specific type of ncRNA, a microRNA, called miR21. What it does is it targets a pathway that basically helps cancer cells survive treatment. So by flipping that switch, miR21 is literally handing the cancer stem cell a bulletproof vest against our best drugs. So here is the bottom line, the really crucial takeaway. These non-coding RNAs, they aren't just involved somehow. They are the conductors of this whole terrible symphony. They're the ones writing the playbook and handing it directly to the cancer stem cells, giving them every single instruction they need to be so incredibly dangerous. Now, just understanding that this playbook exists, that alone is a huge breakthrough. But of course, that's not the end goal. The real prize is to take this knowledge and forge completely new weapons against cancer, to get these incredible discoveries out of the lab
and into the hands of doctors in the clinic. Which, of course, leads to the big, hopeful question, right? If this stuff, this non-coding RNA, is the instruction manual, can we hack it? Can we find a way to get in there and rewrite the instructions ourselves? Can we turn their greatest strength into their biggest weakness?
So how do we actually turn this idea into a real-world strategy? Well, it follows a pretty clear pipeline. First, scientists have to discover which ncRNAs are the key players in a specific cancer. Then they have to validate that in the lab. And from there, they can develop ways to actually detect these ncRNAs in patients. And that whole process leads to these incredibly powerful new tools for things like early detection, figuring out a patient's prognosis, and even tracking how well a treatment is working. And look, this isn't just science fiction. Scientists are using real-world tools to do this stuff right now. Things like RNA sequencing let them scan a tumor's entire ncRNA profile to find new targets. And some of the other tech is so sensitive, it can find tiny, tiny traces of these ncRNAs just floating in your blood. They call it a liquid biopsy. Just think about that for a second. Being able to detect and understand a cancer super early without ever needing to do invasive surgery, it's a game changer.
What this all points to is a truly revolutionary future for how we treat cancer. It's a whole field called precision oncology, and the idea is simple but powerful. We stop treating lung cancer or breast cancer and start treating your specific cancer, tailored to the exact genetic playbook that's running the show inside your body. So here's how that strategy would actually work. Step 1, you take a sample from a patient's tumor and figure out exactly which ncRNAs are calling the shots. Step 2, you develop drugs that can either block those bad ncRNAs or maybe mimic good ones that have gone missing. You're essentially hacking the cancer stem cell's operating system and making it vulnerable. And then, for the final blow, you combine that with existing treatments for a really powerful 1-2 punch.
Now, we have to be realistic here. We're not quite there yet. There are still some pretty big hurdles to clear. Like, how do you get these new drugs exactly where they need to go, to the tumor, without harming healthy cells? And how can we be sure they're going to be effective and safe in the long run? These are the massive questions that scientists are working on around the clock.
But the potential here is just immense. The researchers behind this work really said it best. They said that understanding this whole interaction, it gives us a real path to finally attack the root causes of the most aggressive treatment-resistant cancers. And that's key. This isn't just about a new drug. It's a whole new way of thinking about cancer itself, not just as a disease of broken genes, but as a disease of a hijacked control system. So let's end on this thought. For decades, modern medicine focused almost exclusively on the tiny 2% of our genome that actually builds proteins. But what if the key to defeating our most stubborn, resilient diseases was hiding in plain sight the whole time? In the other 98%, in the vast, powerful, and mysterious world of our genetic dark matter.